Hello and good morning. So, your instructor, Dr. Monica Gulianas again. Today we will be talking about second part of translation. We started working on translation. We already talked about translation initiation and elongation in both prokaryotes and eukaryotes, but I will be reviewing that again today, just in brief, because we will be using some of those concepts in today's lecture. And then I will talk about proofreading and termination of translation. Again, we are talking, this material is covered from chapters 15 and 16 of your book. If you want to read more, you can read these pages noted here. Okay, so translation initiation is it's somewhat similar in both prokaryotes and eukaryotes. We talked about that. Some of the major components of translation initiation in prokaryotes are a 70S ribosome, 3 initiation factors, initiation factor 1, 2, and 3, Shine-Dalgarno consensus sequence, which is upstream of start codon, and this sequence will have a complementary sequence on 16S ribosomal RNA in 30S subunit, the smaller subunit of ribosome, and the modified methionine. The first methionine in prokaryotes is a modified methionine that will be added at the start site. Initiation in eukaryotes also involves all these components, although there are some differences. Ribosomes are 80S in eukaryotes instead of 70S. There are multiple initiation factors. Initiation factor 1, 1A, 3, 4, 5, 5B, and 6 are all required at different steps. Kozak consensus sequence, which is analogous to prokaryotic Shine-Dalgarno sequence, but it is not as conserved as Shine-Dalgarno sequence. This Kozak sequence is a consensus sequence instead. Another difference between prokaryotic and eukaryotic translation initiation is the initiator methionine. In eukaryotes, the methionine is not modified. However, it is carried on a special tRNA, on a methionine FMAT tRNA. Okay, so those are the major differences or major components of initiation of translation in both eukaryotes and prokaryotes. Here it's just to keep in mind, in eukaryotes, transcription initiation happens at the 5' cap, and it also attaches all these subunits. These are the initiation, eukaryotic initiation factor 4 subunits will also bind to poly-A tail through the poly-A tail binding proteins to keep the mRNA in a circle, so that the initiation, so that it all once the ribosomes releases from here, it goes to the 5' cap again for the next round of translation, because the same mRNA can be translated multiple times. Okay, and let's see. So that was initiation. Now let's talk a little bit about elongation. And we've already covered this in detail, so I'm going to talk very briefly now. In elongation, there are 3 steps of elongation. tRNA delivery that leads to accommodation. I will talk about this just in a minute. Peptide bond formation and translocation. So these are the 3 main steps of elongation. What this all means is during tRNA delivery, this elongation factor 2 amino acyl tRNA will bring. So this is your first your initiator amino acid. So this is methionine, or the modified methionine in case of prokaryotes. So methionine is already here. So the next amino acid depends on what the codon is. So the anticodon for that codon will bring the next amino acid, and that amino acid will be brought into this A site. Okay. This amino acid is brought by this elongation factor 2. This 2 has a GTP on it, so this will come here and bind to A site. This GTP will not hydrolyze yet. Once this, once it gets into it, makes this bond with the codon anticodon makes the bond. It's all well identified. This is a complementary sequence. Then this GTP will hydrolyze. Oh, this is FTP. It should be GTP. This GTP hydrolysis will change the EF2 structure, the three-dimensional structure, what it means is this part will stay bound to the mRNA, but this top part will rotate, and this rotation will bring amino acid closer to the existing amino acid so that they can make the peptide bond. So this is called accommodation. So this bringing of this amino acid here is accommodation. And during peptide formation, then the EF to GDP, GTP has already GTP hydrolyzed to GDP, and this has already been dissociated, so that amino acid is now open, now available for making this peptide bond. So during peptide bond formation, peptide transferase enzyme will allow the formation of a bond between amino acid of this amino side of this amino acid and carboxyl side of this amino acid. So these will make a peptide bond together. And third step, which is called translocation, elongation factor EFG, and another elongation factor which is also bound to GTP. This binds to tRNA in the A site. So here in the A site, this EFG will come and bind there, and this binding of EFG to this amino acid will hydrolyze GTP. This GTP hydrolysis will then push this peptide into the exit. So this will exit the ribosome and push this tRNA into the P site of the ribosome. So now the A site is available again for the next tRNA to come and bind to bring the next amino acid. So that's elongation. Keep in mind accommodation, because we will be talking about accommodation in the next few slides. Okay, so now we are moving on to today's materials, today's part of the lecture. So we talked about the initiation and elongation, but what happens if there is an incorrect amino acid comes into the chain? How would the ribosome know? How would the mRNA or tRNA or the ribosome, any of those components, would know that this amino acid is incorrect? So I've already mentioned this, that if during translation, all of the proofreading depends upon the codon-anticodon sequence alignment. If there is a wrong amino acid attached to a tRNA with a different anticodon where it's supposed to be, then it will be almost impossible. It will not. The ribosome will not be able to detect that wrong amino acid. However, a wrong tRNA can be detected because of the codon-anticodon complementarity. If that complementarity is not there, then that will be assumed that this is an incorrect tRNA, so that will be removed from the site. And there are three different methods of the removal of or looking at assessing whether the tRNA is correct or not based on the codon-anticodon complementarity. So the proofreading, it occurs during tRNA delivery before peptide bond formation occurs. So once the peptide bond formation has happened, then that means everything is appeared to be correct and that amino acid will be there. But before that peptide bond formation occurs, there are ways to detect the wrong amino acid, or wrong tRNA. Really wrong tRNA, not amino acid. The first mechanism is site topology. In this mechanism, the site topology stabilizes tRNA and mRNA bonding. So if the tRNA and mRNA are correctly hybridized, then tRNA will remain in the A site. Longer the tRNA stays in the A site, the higher the probability that peptidyl transferase will make that peptide bond when it's activated. So here, if you see in this example, this is UGC here, this is the mRNA. So this AGC is a correct pairing. So this is the anticodon for this codon. So this is correctly bound. Because this is correctly bound, now this whatever amino acid this is bearing, that should be added to the sequence. but this is an incorrect pairing here. Another thing to keep in mind here is wobble hypothesis that we talked about earlier, that this the 5' site of mRNA, 5' part of the codon, is quite important. It doesn't change much. However, the 3' end of the codon has more flexibility. It can wobble because a lot more of these, the codon that's code for the same amino acid have differences in the C terminal of the codon, or 3' terminal of the or 5' 3' of the codon, or 5' of the anticodon. So these changes here are well tolerated, but changes here are not very well tolerated during translation. So this will be an incorrect pairing, and this incorrect pairing will not allow the amino acid or the tRNA to change to make the correct pairing. So here, this is another way to look at it is this is a correct pairing. So this amino acid is brought here. The EF2 bound to GTP is here. This GTP will be hydrolyzed. This hydrolysis of this GTP into GDP will change the will rotate this the tRNA, and it will accommodate. It will bring the amino acid here. GTP will be removed. Amino acid is here, and since this has been in A site for long enough, and amino acid is right there, this will make peptide bond. But if the same happens, if there's an incorrect pairing, so see here, this tRNA is not very well bound to mRNA. So because there is this this gap, this here, this is not staying there very well. So because it's not staying there very well, then it's not very well accommodated, or very well, it does not occupy the A site the same way as the normal well-bound mRNA tRNA will. So in this, it takes this position very well. Here, this is not correctly in the A site. What happens in that case is then this GTP will not hydrolyze. Instead of this hydrolysis of GTP, this whole tRNA will be removed. So no GTP hydrolysis, EF2 tRNA will be released. This will dissociate, and the correct tRNA that has complementarity to this codon will come and bind. Okay, so second mechanism, mismatched codon-anticodon pairs do not occupy the A site in the same way. We just talked about this. This is the incorrect pairing. The delayed GTP hydrolysis also delay accommodation and peptide transferase activity. So there is more time for mismatched amino acid tRNA to exit the A site. Third is accommodation. In this mechanism, because accommodation is important for this amino acid to bind into the last amino acid of the existing chain. So accommodation places the strain on the codon-anticodon pairing, because the codon-anticodon pairing should be correct before accommodation happen. So even if EF2 is able to hydrolyze GTP, for example, in this case, this is the EF2 brought this tRNA here. This tRNA, if it's correctly bound, then it will make this peptide bond here. This is the correct base pairing. But if this is not correct amino acid, even after the GTP hydrolysis occurs, because this is not, the tRNA is not well aligned to mRNA, it will not have the correct accommodation. Correct accommodation means this need to rotate to bring the amino acid close to this this chain here. But if it cannot rotate to bring the amino acid there, then it will not be able to be added to the sequence. So that it will move. It will be released. So if there is incorrect codon-anticodon pair, there is a strong probability that amino acid tRNA will exit the A site during accommodation, because it doesn't really fit the A site as well as the correct amino acid, correct tRNA will. Okay. So now you can see here all these mechanisms, these require tRNA, mRNA binding, the codon-anticodon binding. So it's really, the proofreading is dependent on codon-anticodon hybridization. If the codon-anticodon hybridization is let it go, let it happen. If the if the tRNA fits in the A site well enough and it can accommodate, then that amino acid will be added in the sequence. So because there is no mechanism to distinguish which amino acid is attached to the tRNA, it provides it provides some error in protein synthesis. However, protein synthesis it's still very good. Okay, so that's about proofreading. Now we're going to move on to termination. So because we already talked about initiation, elongation, the next step is termination of translation. So how does the cell know when to terminate the protein synthesis? And in a way, you can say that we have the mRNA has a start site. mRNA also has a stop codon, and then it has the 3' A tail. So that those stop codons, the nonsense codon, and the tail will determine when to stop, right? So that would be a correct assessment, that the stop codon will tell that this is where we need to stop. But the protein, it won't just stop there and the peptide will come out. There are other factors, termination factors, that will help release that that peptide from ribosome. These are called release factors, and let's talk about those. So termination occurs when ribosome encounters a stop codon on the mRNA. Since there is no tRNA for stop codons, so we have 64 codons, right? But there are only 61 tRNA that will bring those amino. There are only 61 anticodons. I'm sorry. So there are 64 codons, but 61 anticodon, because the 3 codons are stop codons or nonsense codons. So they do not have any anticodon. So those will not. So those will be recognized as stop codon. Okay. Termination is facilitated by release factors, or called RF. So initiation factor, elongation factors, and release factors. There are 2 different kinds of release factors, class 1 and class 2. Class 1 release factors hydrolyze the peptide from tRNA in the P site, and class 2 release factors then remove the class 1 release factors from the ribosome. In prokaryotes, there are 2 class 1 release factors called RF1 and RF2. To make it a little more complicated. RF1 and RF2 class factors in prokaryotes, they recognize a different stop codons. These recognize the stop codon via anticodon and bind to the A site. So these release factors are very similar in structure to tRNA, and they act kind of like a tRNA, but they are not a tRNA because they are not bringing any amino acid. These are there to help peptide moved from the ribosome, released from the ribosome. So these class 1 release factors, they recognize the stop codon, and they bind in the A site. So here, this is so it encounters a stop codon on mRNA. So because there is no amino acid, no codon anticodon related to that stop codon, then this release factor 1 comes and bind in the A site of the ribosome. Okay, this RF1 is the class 1 release factor in prokaryotes. Prokaryotes, remember, have 2 of these class 1 factors depending on the stop codon. So for this stop codon, it's RF1. This RF1 comes and bind. RF1 has the sequence GGQ. This GGQ is important. This tripeptide, it moves close to the peptidal transferase center. Here moves here, and this moving of this GGQ close to the peptidal transferase center. The peptidal transferase center is the center that will usually make the peptide bond, peptide formation between two amino acid. But since there is no amino acid, there's not going to be a peptide bond. But it stimulates the center to hydrolyze the peptide tRNA, which is bound in this P site. So this will hydrolyze this, so that way hydrolyze this bond, so this will be able to release, so the peptide will be able to release. So here's the peptide. Now this peptide is released, but the release factor 1 is still associated to the ribosome. Ribosome is still associated to mRNA. So what happens in the next step is this RF3, which is a class 2 release factor. This brings a GDP. Along with the GDP comes and bind to this release factor 1. This GDP then it exchanges its GDP for a GTP. This just shows again, this is the GGQ. It's PF of the release factor 1, and you can see that structurally it is quite similar to a tRNA. This is the tRNA, the CCA sequence. The A is where the amino acid binds, and this is the release factor 1, which because it's structurally similar, it fits in the A site in the same way the tRNA will. Okay, this RF3 GDP class 2 release factor, it binds to RF1 or RF2 if RF2 is present here in prokaryotes, and then this exchange its GDP for GTP here. So this binds here, exchange GDP for GTP, and this hydrolysis, this changes, this helps release this release factor 1 from A site. So the release factor 1 removes as well, it releases here. Now this RF3 GTP is it's just sitting there. Now this GTP hydrolyzed to GDP, and then this releases itself from A site because it is not bound to anything, since the release factor 1 has already dissociated, and since this binds to the release factor 1 and there's no release factor 1 to bind, this will dissociate as well. Okay. This is in prokaryotes. And prokaryotes have two different release factor 1 and 2 of class 1 release factors. This will identify or bind to different nonsense codons, and RF2 is only one. Class 2 has only one release factor, which is RF3 here. Okay. In eukaryotes, there is only one release factor, which is analogous to release factor 1 and 2 or class 1 factors of prokaryotes. So in eukaryotes, there is one release factor of class 1 and one release factor of class 1. So these are called eukaryotic release factor 1 and eukaryotic release factor 3. This release factor 1 in eukaryotes recognize all 3 stop codons. And here, this is a really nice paper. If you want to read more about it, what I really want to highlight in this paper was the termination of translation in bacteria, so in prokaryotes, require 2 release factors. RF1 responds to the UAG and UAA stop codons, and RF2 is specific for UGA. And also recognize UAA. So UAA is recognized by both UAG is by RF1 and UGA is by RF2 in prokaryotes. In eukaryotes, it's universal, just the eukaryotic release factor 1 responds to all 3 stop codons, so keep that in mind. Okay, so now until now we talked about, okay, so the peptide is removed, there is nothing in the A site is free, but the mRNA is still bound, the ribosomes are still bound, so these need to come apart as well for termination to completely be over. So the final step of termination is the removal or disassembly of the ribosome from the mRNA. And this is carried out by another factor called ribosome recycling factor, RRF. This RRF is, again, it's structurally similar to tRNA because it's going to bind to those sites in the ribosome, so they need to fit in the site, so they're structurally similar to tRNA as well. It promotes dissociation of the ribosome from mRNA and stays bound to the small subunit. So here, let's see. So this is mRNA. This is our small subunit and large subunit of the ribosome. This tRNA, it's still bound here in the this is the exit site, this is P site, this is A site. So this tRNA is because it's an exit site, it's going to release anyway, but this will be pushed out. The mRNA and these ribosomes need to come off from each other. So here, RRF, or ribosome recycling factor, this mediates comes and bind here in the A site. This is the EFG or RRF complex here. This binds together in the P site and A site. This changes the conformation, leads to GTP and GTP hydrolysis as well here, and this. Because of this hydrolysis of GTP, there will be transient separation of subunits of ribosomes. So here the ribosome subunits are separated transiently. That means these they can come and join together. But at this point, these are, so mRNA is released at this point. Both these factors, RRF and EFG, these come off of the ribosome. The ribosomes are now separated. Initiation factor 3, remember the function of initiation factor 3 is to keep the subunits apart, so to remove those. So this initiation factor 3 binds to small subunit and keeps those subunits away. Once everything is dissociated, all these extra components are dissociated, IF3 is also released, and then these two come and bind again. So you will see that as when we started initiation, we started with the dissociation of ribosomal subunits. So ribosomal subunits are together. When the translation initiates, these need to come apart. These need to move away because the whole machinery aligns on the smaller subunit, and then a larger subunit has to come in place later. So this is now ready for the next translation event. Okay, so let's also talk about, if we talked about proofreading, well, let's talk a little bit about translation and mRNA stability. So what happens if the mRNA is corrupted, if mRNA is not a correct mRNA, or if mRNA has a mutation in it? If the mutated mRNA lacks a nonsense codon, if there is no stop codon, how the termination will happen in that case? Because we talked about this stop codon. The stop moving to the stop codon leads to association of all these different factors, these release factors, and then the dissociation of the ribosome. But if there is no stop codon, then how would the cell know that this mRNA has a problem, and then it needs to be degraded or removed? Okay, so that's what we're going to talk about now. So the mRNA that lack nonsense codon or stop codon, the ribosome will stall with the mRNA 3' end in the P site. So here in this example, if you have a 3' end broken mRNA, this mRNA doesn't have 3' end. So one thing to keep in mind that with the 5' cap of mRNA and the poly-A tail of the mRNA really provides them stability. And we talked about this earlier, that if either of these are removed, then the mRNA will not be stable and it will be degraded fairly quickly in the cell. So that can also affect the gene expression of mRNA. If it lacks 5' cap or 3' poly-A tail, mRNA will be broken down. It won't be stable, so that means it won't be able to express. Here, if it makes it to translation and it reaches to the end where then there is the 3' end is broken, in that case, because there is no poly A tail, right? So it's broken. Or think about it in prokaryotes, because prokaryotes don't have a poly A tail or 5' cap. So if this is a prokaryotic mRNA and it reaches to the end, there is no stop codon, and so the translation has reached towards the end. Now what happens in that case is there are the small stable RNA A, or also called a tmRNA. This is in short SSRA RNA. This SSRA RNA enters into the stalled ribosome at A site, because there is nothing for the ribosome to do here. There is no real signal to stop the step translation. So this is the last codon. It's here. It's just sitting there. So it will this SSRA tmRNA will enter into A site. This SSRA tmRNA, this has alanine, tRNA bound to alanine here with the EF2 GDP. So this SSRA tRNA binds alanine and it's entered the A site complex, and you can see that it also has, because it's an mRNA, tmRNA, termination RNA. This termination RNA has its own sequence and its own stop codon. So this will enter into the sequence. Okay. Once this enters into the sequence, it will complex here, and the complex with the F2 GDP. It's gets here. This GDP, EF2 GDP will be hydrolyzed. This alanine will make a bond with this peptide bond will formed here. There will be okay, let's go to the next slide. So I was going to talk about transpeptidation, but let's talk about that in the next step. So this once this SSRA RNA binds tRNA alanine, enters into the A site, it's complexed with the F2 GDP, and it eventually replaces the mRNA in the ribosome. So it will this mRNA is here. It will do the transpeptidation, so it will switches, and then it will this will be used as the template for the rest of the mRNA. The translation of this SSRA RNA ORF, so this portion right here, translation of this ORF will yield a C-terminal tagged protein. So this protein now will have this C-terminal tag. This, because the cell already knows this, if there is this at the 3' of the protein, if this is the sequence, then this is a wrong protein, or this protein will be labeled for degradation. This SSRA RNA, it will also stimulate RNA degradation, so all the mRNA will be degraded, so it will not make the next into the translation machinery. So here again, this is the stalled ribosome. There is this 3' end broken mRNA. This termination mRNA binds to tRNA that carries alanine along with EF2 GDP. It will enter into the A site. Once it's recognizes by this recognition by SSRA RNA, it means it enters into here. This SSRA RNA will then this EF2 GDP will be hydrolyzed, and this alanine will be presented. So this is accommodated here. Transpeptidation happens, so these peptides will switch here. So these peptide switch here. So alanine is here, and the one that was there switches the form, and then it uses from here. It will keep on moving. It will use these codons on this termination mRNA as the rest of the protein synthesis. This is translocation and replacement of mRNA. So here, this mRNA is still mRNA is there, but this mRNA is already translated. So now this is this part of the mRNA that needs to be translated, right? And here, this will continue translocation of mRNA reading frame. That means it will continue adding the amino acid based on the codon. So it doesn't switch the codon; it still keeps the codon, and then once it reaches to the stop codon, then it will be the similar way of termination as it would be if it was a normal mRNA molecule. However, this protein is now tagged. So this is a tagged protein, and these tagged proteins they then are broken down by or degraded by the cellular proteases, because cellular proteases recognize these sequences and they know that if there is this on the 3' tail, then this need to be degraded. Okay. In eukaryotes, if the protein is produced from an mRNA that lacks a nonsense codon or a stop codon, then they will have a poly K C-terminal tag, because eukaryotes have the poly A tail in mRNA. So if they miss the stop codon, the translation will go all the way into the poly A tail. Usually, as you know, that the stop codon comes before poly A tail, right? So if there is no stop codon before poly A tail, then the poly A tail will be translated as well. And since AAA codes for lysine, so all those 3' towards 3', it will be lysine, 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 so polylysine. Those polylysines also then trigger the cellular proteases, because that's not normal for proteins, so these will be deleted, or those will be degraded. The ribosome stall at the end of poly A tail. So if there is no stop codon, it will go into poly A tail. This poly A tail will be the lysine will be added multiple times, because poly A tail is around 200 to 250 nucleotides. So that three times of that will be about 65 to 70 amino acid will be lysine amino acid. So once it reaches to the end of the tail, at that point the tail will attract these proteins called DOM34 and HBS1. This DOM34 and HBS1, this binds to GTP. Again, they look very similar. They're structurally very similar to tRNA, structurally very similar to release factors. So this will come and bind here to the A site of the ribosome. And the binding of this DOM34 and HBS, this will promote ribosome disassembly. So the ribosome disassembly, because ribosome disassembles, then this protein will come out, the peptide will come out as well. SCI7 is another factor, another protein which stimulate the ribosome release. So this will dissociate these two DOM34 and HBS will disassemble the ribosome. SCI7 will make them release this mRNA. Okay. Now, because there is poly K tail here, so these poly K tail will be recognized by the proteases. Because these proteases know that if there's multiple lysine, this is not a correct protein. So this will be then lyased. This will be degraded by these proteases. If there is a premature nonsense mutation, that means if the stop codon enters itself somewhere in the middle of the gene, middle of the mRNA, then the SR protein, those serine arginine rich proteins that we talked about, they present in both introns and exons. Because there is no intron at this point, so the SR proteins that are there from intron splicing in the exons and the elongation release factor 3 in the ribosome, these will attract a protein complex with RNase activity, and these will either remove the 5' cap or they will remove the poly A tail. And removal of either of 5' cap or poly A tail will again stimulate the production of this endonuclease, or these endonucleases will come and they will degrade the uncapped RNA, or RNA that doesn't have a poly A tail. So here in this exosome, this is removing the one that doesn't have poly A tail. This is example of the one that's removing the one with that doesn't have 5' cap, protease that recognizes the protein with poly K tail. So these are different mechanisms, different ways of termination of termination of translation in normal proteins as well as in proteins that or mRNA that might be mutated. How the cells get rid of those proteins. And with that, this topic of translation is over, and we will move on to the next topic on Monday.